Hello everyone, my name is Darla Saunders and on behalf of the Atlantic Salmon Conservation Foundation and the Canadian Rivers Institute, I would like to welcome you to today's webinar. This series is made possible in part thanks to the contributions of the Government of Canada. For today's webinar, we're very pleased to be hosting Lindsay Brin. She's an ecosystem ecologist who has spent over a decade conducting graduate and postdoctoral research in the fields of biogeochemistry and microbial ecology, giving her a broad skill set in the management and analysis of ecological data. She's currently a postdoctoral researcher at the University of New Brunswick's Canadian Rivers Institute, developing a project to use aquatic vertebrate community data to make predictions about whether ecosystems have been impacted by disturbance. She's a certified data carpentry instructor and is also involved with the Ladies Learning Code. After the presentation, we'll be opening up the floor for a question and answer session. You have the option of asking questions directly using your microphone, or you can type them in and we'll read them aloud. I will now turn the webinar over to Lindsay.
Hi everybody, thank you for joining me today. Today I'll be talking about data analysis with R. I'll introduce you to the program and the environment that you can use to work in, some basics to using R, and also give you a sense of what possibilities you have down the road in terms of structuring your analyses. There are a couple of different objectives I have for the next hour. The first one is to give you all an understanding of the benefits of reproducible research and using a coding or computational programming approach such as R, but R is not the only one you could use. I also want to introduce you to the workspace in terms of R and RStudio and the associated workflow so that it becomes familiar and more accessible to you when you try to put it to implement it yourself. Third, I'll be presenting you with an overview of the types of data manipulation and analyses that become feasible within this environment. And last, I'd like to point you all towards some resources to learn techniques and analyses in more detail. So clearly, with 1 hour in a less interactive format, I'm not going to be able to get you to the point where you can do all of your analyses in R right away. But what I'm hoping to do is sort of build a solid foundation and a sense of accessibility. So some of those barriers to changing the way you approach something will be a little bit lower, and hopefully will make you a little bit curious about how you can switch this approach. My basic thesis in the next hour will be that computational programming can help make your analyses more efficient and reproducible. I just need to minimize something really briefly. Okay. So your first question might be, why should you use R for data analysis? And this main question is really made up of 2 sub-questions. The first one is, why should you use a computational programming approach in the first place? And then the second one is, if you were going to use a computational approach, why would you choose to use R in particular? So I'll start with the first question. The biggest advantage that you get from using a computational programming approach is that you move your work towards reproducible research. So all of your analyses are written out and documented, as compared to working in an Excel file where your analyses are sort of hidden in the references between different cells. When you're working with a script, when you're doing computational programming, all your analyses are in one place and written out and documented. Also, I'll go into some of the more of the details between the structural differences between the more typical Excel or spreadsheet-based workflow and computational programming. But one of the main advantages that I'll show you and talk about is that you can avoid altering your original data files. So instead of working within that original data file and having to make sure that you don't accidentally type over a cell that has data that's important or delete something, you save your files, you don't touch them, you can read them and work with them and not change the original files. And so that gives you sort of a barrier and some safety, which I myself have found extremely useful. Another advantage is that this approach makes it simpler to rerun analyses, whether you're checking your work yourself, if you just want to check your stats, if you're trying to give your project to somebody else to check over it, or if you're extending your analysis to something further in the future. So a couple of things that includes that I want to highlight is that this approach makes it easier to prevent or spot mistakes. So because all of your analyses are in one place, you can check them over more quickly. And also you can, as I said before, keep from altering those original data files, which is great. Another thing is that it makes it easier to change or extend analyses. So if, for example, you change your mind about how you want to do like a standardization or how you're running some sort of model with a log fit or a linear fit, you can just go right to that one part where you have done your analysis. And if you've structured the whole analysis well, you can make one change in your code and apply it to everything downstream rather than having to sort of hunt through different files and change them all in all of the relevant places. So R has some advantages itself as well. One of the first ones is that it's free, and I mean that both in terms of the cost and also in terms of it being unshackled. So it is free to use, it doesn't cost anything, and it's also open source, so people contribute to it. It's an open source project that's constantly changing, and this has a lot of benefits. It's also well documented, so if you run into problems, you can often find answers or solutions to your problems online. It's cross-platform, so if you yourself are thinking about switching between Mac and PC, or you're working with other people who are working on a different operating system, you can still use the same program and the same code with some very small changes that have to do with working with your original file system. But most of the functionality of R is exactly, it's not only functional on both platforms, but it would be exactly the same on both platforms, which is great for collaboration. There's also an active community of R users, especially within scientists. So the best part about this is that community members develop packages that can expand the functionality of base R. So if somebody has come up with a different way of analyzing some kind of data, whether it's spatial data or sequencing data or what have you, they can bundle those functions together, maybe with some sample data sets, into something called a package. And then those packages are freely available to be downloaded. So sometimes you might be reading a paper about some analysis, and in the methods it'll say, this analysis was done using XYZ package in R. And then you can very easily just install that package and pick up the same tools that they use to be able to apply those analyses to your own work. Another reason this active community is really great is that there are a lot of community resources for getting help. At the very end of this talk, I'll put up a slide that explains some of those resources, but really what it comes down to is if you have a problem, if you Google your problem, there's a pretty good chance you'll find someone who's had almost exactly the same problem and other people who have given answers for it. So this is really great for that sort of faster paced, quick learning because there are people around you to help. R is also at least as good as, if not better than, commercial alternatives. So there are other things that you can use like SAS or Systat, but they generally are more expensive. There are other free programs that are similar. The other one that's used mostly in science would be Python. And there are a lot of really vociferous debates about whether R or Python is a better approach. Honestly, anything you can do in one program, you can do in the other. It really just comes down to what the community of people around you are using. And that's, if you're making a decision between those programs, that's really how you should make the decision. Last, R is a useful tool for many aspects of research. So through your whole workflow, you may find it to be useful and help you do your work more efficiently. Okay, so there are a couple of structural differences from working in spreadsheets that I want to start by going over. With a typical approach, you have a lot of different kinds of data in the same spreadsheet. So I'll be talking about two different fake experiments that I've put together as examples throughout this talk or webinar. The first one is this fish analysis. So theoretically, someone went and sampled a bunch of different fish, sticklebacks, in St. John Harbor, Kennebecasis, and this great site aptly called Contaminated River. And in those sites, at those sites, people have sampled fish and measured the length, and then taken their tissue and done some analysis for total mercury and arsenic, and then done some analysis with the mercury. So often if you're analyzing this data within one spreadsheet, you might have a separate file that has the analytical analysis off of an instrument from the lab, and also another one with metadata, but you'd end up putting them all together in one spreadsheet. So you've got your original data, which would be like your sample ID and site and data, metadata. And then you've got your organized data, so maybe some sort of standardization. And then you have your analysis. So here I've got a pivot table that's looking at the mean and standard deviation of the mercury, the corrected mercury number. And then you might have a bunch of figures, and maybe they'll be on different sheets within the workbook. but often they're all kind of in the same place. If you're working with R, you start off by structuring your files in a different way. So hopefully when you set up, when you downloaded the files that I sent out, you were able to set something up that looks something like this. So first of all, I mean, it's a good idea, no matter what your project is, it's good to have one project folder, and that folder is called your working directory. So here I've got a folder called Intro R Online Course. That's my working directory. Within that directory, I have a bunch of subdirectories. One is for data, that's where my original data goes. I've also got two output folders for data and figures, which I'll talk more about in a moment. A scripts data for the R, or scripts folder for the R scripts, and then a documents folder, which is where you can organize all of the rest of your related documents that aren't analysis related. So metadata or proposals or presentations or manuscripts or what have you. In that data folder is where you would put your original data. Usually you'll use CSV files because they're easiest to read into R. So here you'd have your fish contamination, output from the analytical instrument, and then your metadata as two separate files. And there's no need to put them together manually. You just have your separate files in the data folder. You then have a bunch of scripts in the script folder, and R scripts are basically your recipes for analysis. So they're basically just text files, but they're text files that are made up of commands that R can read, that are recipes for doing the work that you want R to do for you. Those R scripts will often have outputs. One possibility is that you'll have organized data, so that's your output of scripts that have to do with pre-processing that you'll do with your data that you might want to look back at later. And then you might also have figures as your output of these scripts. So I often output my figures directly to PDF so that they're easily formatted for publication and easy to put into presentations or anything else. And those you want to put in a separate folder, which I usually call figure output. So one key thing to realize here is that if you have this sort of folder directory structure, anything that goes into your data folder, you don't touch, so it's basically safe. Like, you don't delete it, you don't move it around. You know that you need to have it there for safekeeping. And then anything that you put in your data output or figure output folder is something that you can generate with your scripts. So you don't have to worry about deleting a file or changing something or anything else, because it's all just easy to recreate and regenerate. Okay. So the next thing I want to do is introduce you a bit to RStudio and working within it. RStudio is, it's called an Integrated Developers Environment, Development Environment, or IDE. It's basically a graphic user interface. It's an environment that you can work in that will help you organize your work within R and on your computer generally. You don't have to use it. You can use R directly, but it just makes things a little bit easier. So I'll switch over to RStudio. If you're following along on your own computer, you can open RStudio now, and when you open it up, you should see something like this. So on the left side is your R console, and we can start by opening a new script, a blank script. You can do that at the top left of your screen. There's this white rectangle with the green and white plus sign. You can click there and then go to R script for new R script. You can also go to the File menu at the top of the screen and go to New File and then R script, and that will do the same thing. So at this point you have 4 different panels, and you can resize the panels however you want to constantly. On the bottom right you have a panel that has a bunch of different tabs and different options. So the first one that I want you to look at is this Files tab. This is basically like an Explorer, a file explorer, so it's the same. I'm on a Mac, but it's the same as if you were going to move through your files on your own computer, except that it's just handily packaged within one program. So what I'd like you to do now is navigate to your project directory, your working directory. So I've got mine in Short Intro to R is my project directory, so I'll click on that. And then once you're in your project directory or working directory, you want to set this as your working directory. So you're basically telling R, this is my reference point for all other file names. This is where I should go back to. This is like the home project folder. So to set it as your working directory, you go up to this little button that has gears and says More, and then go down to Set as Working Directory, and click that. And you'll see on the bottom left in the console a command that was entered into R that will appear and will have been run. So now you have set this as your working directory. Okay, so I'll talk a little bit more about the console, which is in the bottom left. At its very basic, your console can be like a super hyper overpowered calculator. So you can do really simple math in it if you want to, and you can just try a couple of math problems here. You can do multiplication, division. You can use parentheses. Great, not all that exciting. Things to notice. When you enter a command, or when you look at the console, you'll see a little caret here on the bottom left, and then a blinking cursor. That caret means that you can enter in a command, and then R is ready to take instructions. And then after you've entered your command, you'll see this square brackets around a number 1. That means it's your first result, and then your answer is there. It's great that you can do stuff in the console, but anything you do in the console sort of disappears afterwards. The R console doesn't have any memory. So, I mean, I can press the up button and I can go back to previous commands, but this doesn't mean anything moving forward. If I opened another instance of R, or I closed it and opened a new session, it wouldn't be anything that would be remembered. So this is where scripts come in. So in the top left here, you've got your R script. So far it's untitled, but you could save it. And the reason that scripts are really powerful and really useful are because you can run code from them. You can send commands from the scripts to the console, but you can also save them for later. So you can set up a complex analysis for how you'll move through your data and analyze your data. And then you can open it again down the road and run the same analysis without having to retype everything back in again. So in this new script, I'll just show you the same basic math and show you how to run it. So do the same calculations. Okay, so when you're working in a script, if you want to run some of that code, there are a couple ways you can do it. One is that you can just put your cursor on that line and then go up to the run button here and click run. And what that does, you can see in the bottom left, is it acts as if you had typed that command directly into the console. It just gives you the command and then the output. You can also highlight just a bit of the code. So if we wanted to run just the first part of this calculation here, we could highlight just 3 plus 5 and then click the run button, and it'll only run what you've highlighted. If you want to run the entire script, you can click the source button, which will source your file, and it will run all of the calculations that are within that file. Of course, this becomes a lot more exciting and more powerful when you're doing things other than adding 3 and 5 and dividing by 2. If you want to save your file, you can click on this little old school floppy drive to save the current document. There's one here, there's one here, and then you can also go up to the file menu. I don't know why it's so redundant. So you can save it and then save that file wherever you want. I'm not going to save this one in particular. So I'm going to move on and talk about some of the other panels that you have, or panes that you have in R. In the top right, you have 2 panes. One is your environment and one is your history. Your environment tells you what objects you have saved in R. I'll go back to that in a little bit when we're working with a sample script. Your history shows you what recent commands you have run, which is helpful if you're looking at the output of something and you want to just double check what the command you run is, but I generally find it isn't extremely helpful because you're not going to scroll up for 300 lines to figure out what your first analysis is. So generally save things in your scripts and don't depend on your history. The bottom right pane has a whole bunch of different options. The file one we've already worked with briefly to navigate to our project folder. The plot window, we'll see a little bit later, that's where plots will come up. Packages, I talked a bit about packages. That's when people bundle functions and other useful data together and then can distribute it. You should all hopefully have been able to download the dplyr package. If you've been able to download it, it will show up, or to install it, it will show up as a package in this list. And I'll talk a little bit more about using packages later on. You also have a help pane. help pane is where you can look up information on different functions that you might want to use, or just any word in all of the help files. So that will end up being often quite useful down the road. So we could say, let's look up how to round something, and that will give us the function for how to round something. The viewer is basically for web content, so you are unlikely to need to use that anytime soon, and I wouldn't worry about it too much. Okay, so the next thing that I want to do is run you through 2 example scripts. Basically, I'm going to use this to demonstrate some basic concepts in R and get you to get some experience creating objects and doing some simple manipulations. But also, I wanted to use this as a way of demonstrating what a workflow could look like if you move your analysis to R. So data management and analysis, how to use statistics, and also a little bit about figure output. I recognize that I'm going to have to move through these kind of quickly, and I won't be able to give you the practice that you would really want for each of the individual concepts. But I'm hoping that this is a way to give you a broad overview of what you can do, so you have a sense of why you might want to take the time to learn how to do it and where this could take you to. So there are 2 different scripts that I'll walk you through for 2 different fake studies that I have created for the sake of examples. The first one is fish contaminant analysis, which is the one that I am showing you the project structure for. And that's basically a focus on sample collection and lab measurements and also some statistics. And the second one is a nitrification calibration script, which will kind of show you how to do some calibration from a laboratory analysis, but the basic focus there has to do with how functions can be useful and why that would be a great skill for you to learn, or tool. And then second of all, how you can output files and also switch from one data file to another, or like apply your analysis to a new set of data in a way that will make your work more efficient. So ideally you have these scripts saved into a scripts file. You should have nitrification calibration and fish contaminant analysis, and we'll start with fish contaminant analysis. There are a couple of different ways that you can open this in RStudio. If you double click it and just open it, it will probably open in R and not in RStudio, which is fine if you want to go that route, but I'm going to demonstrate how to do it in RStudio. So you can right click and go to Open With and then go to RStudio. Or I know this works on a Mac, and I don't know if it works on a PC, but on a Mac at least you can take your file and drag it to the RStudio window and it will open up. Okay, so here's an example of a script that might be the kind of analysis that you might find useful down the road. There are a couple of different things that I want you to notice in this script, sort of as an overview. One is that there are a lot of lines that have these hashtags or pound symbols at the beginning of this line. This indicates a comment. So any command that is sent to R where there is text to the right of this hashtag will not be evaluated. It's technically run, it's just not evaluated. So if I highlighted this line and I clicked run, you can see that it appears in the console, so it was sent to R, but it's not evaluated as anything. So I could theoretically type a math problem 3 + 5, and then highlight that and run it, and the command is sent to R, but it doesn't give me the answer 8 because it's not evaluated. So I'll delete that again. So those are comments. Comments are extremely useful and a very good practice to get into. It's a good idea to start all of your files with a header that has a bunch of comments. So there you can put information like the file name, your name, the date, and what your file does. And that way, if you were to ever look back at this script down the road, if you're redoing an analysis a couple months later, or you've gotten comments back from reviewers on a publication and they say, actually, I'd like you to change your analysis, then you can go back to your script and actually remember what you did. Do not underestimate your ability to forget how you did an analysis, even like a week ago. Another reason it's good to have this file name in here is that if you were ever to print out the script, then you'd be able to figure out what the file was. So yeah, so start with a header, start with information on what the script does, who wrote it, when you wrote it. I usually start with a start date because it usually takes me a couple of days at least to write a file. as I think through how I'm going to change my analysis. You know, it's something that evolves over time. Comments are also really useful throughout your file, throughout your script, so that you can annotate each of the commands, each of the functions. I actually often start by writing out what I want to do in plain English with those comments, like, I'm going to read in these files, I'm going to join them based on this key, I'm going to do an ANOVA to look at whether there is a significant difference between these different groups. And then if you start by those comments, that kind of helps you structure how you're going to write out your analysis downstream. Yeah, so comment a lot. I cannot overemphasize how important it is to put comments in your data. Another thing that you might notice in this file is that there's this funny little symbol here that looks kind of like an arrow that's a caret and a dash. That's called the assignment operator. That's one of your really basic tools in R. So let me take a second to go into assignment. In the console, you can follow along. I can demonstrate assignment by assigning the value 3 to the variable x. So if I type something like this, x left caret dash 3, that means I'm assigning the value 3 to the variable x, or I can read it as 3 goes into x. If I hit enter, you can see that in the top right of the screen in my environment, there's now a variable called x with the value 3. So basically, you can do lots of math in the console, but you can't get very far until you start creating objects. This is the simplest kind of object. It's just a variable that has the value of one number, but you can start to create much more complex objects. You can create objects that are, instead of numeric, that are string characters. So I can say y is, and then if I put something in quotes, it's a string, a character string. So I can say my character string. And now I have an object called y that has this value, my character string. You can look at the values of objects by looking at them in the environment, or you can send the name of the variable to R as a command. So I can hit X and hit enter, and it will tell me X is worth 3, or Y, and it will tell me Y is worth my character string. You can call variables or objects X, Y, Z, whatever, but you can call them also long words or anything else you might want. So I can say my object has the value of 458. And now I've created another object with that name. I could name it Constantinople. I could name it supercalifragilisticexpialidocious. But I recommend that you name your object something that is reasonable, makes sense, and will indicate to you what that object actually is for. So yeah, so that's the assignment operator. One other thing to notice consistently in this file are that there are things called functions. So when you see text followed by a parentheses and then something inside that set of parentheses, that's a function. So here, summary is a function, head is a function, going down, bright.csv is a function, box plot is a function. Functions are basically little scripts that help you do useful things. So it's just like any other command that you might type into the console, except they're all bundled together. So for example, let's play with the function round. So to use the function round, we just type round into the console. Let's give it the argument of the value of pi, so 3.14159. And if we hit enter, what that function does is it's rounding the value of pi to the nearest whole number. But you may think, well, I don't want it to round to the nearest whole number. I want it to round to 2 significant digits. How does this function really work? And there are a couple of ways you can look at that. One of them is to look at the arguments of the function, which is the details of what you're telling the function to do. You can do that by running a function on a function. You can say, args of the function round. And that will tell you that round is a function with 2 arguments, x and digits. We can also get more information on the function round with a question mark. So you can type question mark round, and that will pull it up on your help file in the bottom right. So we can see from the help file that round rounds the values in its first argument to the specified number of decimal places, and that the default is to use 0. If we look at this usage line, we see what we saw from the args argument, which is that the function round has two arguments. One is x and the other one is digits, which is set to a default of 0. So this digits equals 0 means that if you don't tell it how many digits you want, it will automatically use 0. And then if we go down, we can look at the details of the arguments and see that digits indicates the number of decimal places or significant digits to be used. So that gives us information on how to use the function round. So there are two arguments to the function round. There's the x and there's the digits. So let's try giving it a different argument or different value of the argument digits. So we can say 3.14159 and then tell it digits equals 2. And if you run that command, you get a value back of 3.14. So this first thing here, 3.14159, is an argument, but I haven't specified what that argument is called. So if you put the arguments in the order that they're defined, so x and then digits, you can avoid using the names of the arguments and just type them in. That will give you the same answer. Alternatively, if you name all of the arguments, you can put them in whatever order you want to. So I can say digits equals 0, x equals 3.14159, and let me try digits equals 2. And I'll get the same answer back. But for clarity, as you're moving through your files and trying to remember what each of the function does, it's generally a good practice to put the required argument first and then to name all of the rest of your arguments. So I would typically write round 3.14159, comma, digits equals 2. And that way there's a lot less confusion about what you're trying to do with your function. So you can play around with that function, you can play around with other ones. There are some really, some of the ones you might expect, like min or mean or max, but also lots of other ones that will help you work with data in R. Okay, so what I'm going to do now is walk you through the fish contaminant analysis.r file, and then walk you through the other file and hopefully give you a sense of the some broader processes that you could follow while you're working with your data. And again, apologies that I'm going to be going kind of quickly, and I'm sure some of the lines you won't be able to deconstruct and take apart. And if you'd like to get more information, I would invite you to come to the workshop that I'll be teaching in January over 2 days, which I'll tell you more about later. But in the meantime, don't worry too much about the details. The idea is to get a broad overview of what you can do with R. Okay, so if you were successful in installing the package dplyr, it should have showed up in your bottom right tab, the Packages tab. You can search for it. You should be able to see it there. If you have not been able to install it, you can try to do that right now. You can use, the install packages function and give it the argument dplyr in quotes, then do a comma and say dependencies equals true. If you've already installed it, don't worry about this. It means you've already done it. If you didn't install it or you had trouble with it, you can try it again. And if you run that line, you should see it will connect to the internet, it will download the package, and it will save it in your installed packages. So at this point, and I should say for a moment that when you see red text in R, it can either be an error or a warning. If it's a warning, it'll usually say a warning. If it's an error, it'll usually say an error. So just seeing red text doesn't necessarily mean there's a problem. So in this case, it's telling you what it's doing, and it's either a warning or just a message. Don't worry about it unless it actually says error and doesn't do what you want it to do. So installing a package is kind of like going to the store and buying a bunch of tools. And if you're working at your work desk and your new tool is in its box on the ground next to you, you can't work with it yet. So once you've installed that package, you need to then load the package into your current R session. And that's kind of like taking your tool out of its box and putting it on the desk so you can now work with it. So even though you only need to install the package once on any computer, you need to load the package into R every time you want to use it, every R session that you want to use it. So I'll load it in now, and then I'm good for the rest of the time that R is open. But if I quit R and then I open it back up again, then I would have to reload that package so that I'm like putting the tool onto my table to be able to use it. So if you're writing a script that you want to be able to be a standalone script, one of the first things that you want to do is to load the required packages. So let me back up and say this R script has been written to read in data for this fish contamination study, join the contaminant analysis data with the sample data, standardize the contaminant data by fish length, and then assess differences by treatment statistically. So that's what I'll be going through in this file. Okay, so if you haven't loaded the package, you should run this line library. It's a library function, and it loads the package dplyr. Note that dplyr is not in quotes. So if I run that line, I'll get some information from R. It says attaching package dplyr. It also gives you some information on different function names that it has redefined. Mostly you don't need to worry about that too much. As you start using more packages, it might start being relevant. The next thing you want to do is set your working directory. So if you've already done this manually, you don't have to do this again in the script, but it's still a good thing to put into your scripts so that your scripts can stand alone functionally. So this is saying, okay, R, this is where you are. This is what everything else that I read in will be relative to. So set your working directory. The next thing that I'm going to do is read in data. So here I'm using the function read.csv. I'm giving it an argument that is my file name and where it is saved. If you have a file, if you have a folder called data and you have the file name that's exactly the same as what I sent out, you should be able to run this line as long as you've set your working directory to your own file system. You probably don't have the same file system as me. So when you run this function read.csv, it reads that file and then it assigns it to a variable called fish metadata. So I'll run that in the next line. And now I have in my object two sets of data, fish contaminants and fish metadata. I can look at them in a couple different ways. I can just click on one of them to open up in the R viewer. I can also run that line that's the name of the variable, and that will show me in the console the value of that variable. So here we've got fish metadata, and you can see that it's got the same thing that was in the CSV file, which is a bunch of different variables, which are the columns, and a bunch of different observations, which are the rows. So now I've got my fish metadata and my fish contaminants. There are a couple of ways I can look at that file, those data sets. One of them is with the summary function. If I run this line and pass the fish metadata file or variable to the summary function, it will give me a bunch of different information about each of the variables in that data file. So here are the categorical variables. R will tell me with the summary function what variables or what observations exist in each categorical variable. And then if you have a numerical variable, it will give you information on the mean, median, mean, max, and the quartiles. You can also use this function head to look at your data, and that will give you the first 6 lines of your data frame. And so that's not going to give you the whole picture of the data, but it's often sort of an easier way to visualize what the structure is. You can do the same thing with the analyte data, summary, and head. Okay, so we now have 2 data sets. One of them is fish contaminants, and the other one is fish metadata, and both of them have this variable called sample ID, which is NB1 through, I think, 30, 24, 30. And what we want to do next is join the 2 files so that each row is one sample ID, and we add the information on total mercury and arsenic to that metadata file. So this is something that, if you're using Excel, can take a little while because you have to make sure that you've got your data in the right order and that you don't do an accidental referencing mistake, or you can use VLOOKUP or something, but I don't know. I found that it often ran me into problems or kind of was clunky and took a while. So here all you need to do is use this one function called left join. There are different kinds of joins you can do. This one happens to be a left join. That's all in the dplyr package. So I'll join fish metadata and fish contaminants by the key, which is sample ID, and then call that fish data. So I run that one line, and then look at the top of that file with head. And now I've got a data frame that gives me all of that information from the two different data frames put together, correctly identifying which sample ID from one file should be joined with the sample ID with the other. So again, this is a way of sort of avoiding those manual mistakes you might make by just accidentally clicking on the wrong cell. The next thing that I'm going to do is create a new variable called total mercury standard, or standardized, which will be total mercury divided by length. And this will use the mutate function from dplyr, that package that you loaded earlier. One thing to note here is that this does not affect the original data file, and that it's really easy to then go in and change how you do this calculation. So again, if an R script is sort of your recipe for your analysis, all you need to do is change the recipe to do it differently next time. So we'll run this line, and then if we look at the head of fish data again, it now has another column added to it, which is total mercury standardized. If you decided later you want to standardize by weight, add another variable, or you want to do by, I don't know, half length, if that was important to you, all you'd have to do is change that one line here and change your analysis. Once you've got organized data, it's a really good idea to write the full data set to a CSV file so that you can then read it in later, and we can do that with the function write.csv. So going back to the file structure, we've got the data output folder here, which is empty as of now. And if you run this write.csv file, assuming your working directory is set up properly and assuming that you have a data output folder, you will end up with a CSV file that has that organized data with all of your information joined together, and also your standardized mercury variable as a variable. So you can kind of start to see how if you wanted to apply the same sort of thing to another set of files, it wouldn't take as much work to then just take the same code, change what your file names were, and organize your data in the same way. Okay, so now we want to look at the actual data and look at how total mercury varies by site. The first thing that you should do generally in any analysis is look at your data visually to get a sense of whether there is an analysis you want to do statistically. So we'll start out by looking at it with a box plot, and that's just a question of using the box plot function. This first part of the box plot function is really all you would need to make a box plot, and then this latter part controls the aesthetics of the plot. So we're adding a Y label, we're controlling the Y limits, the limits of the Y axis, and also something that has to do with the rotation of the Y labels. So run this one line, and then the bottom right, you can also zoom in to look at that figure. We now have a figure that shows 3 different samples and information on the mercury concentration for the samples from each of those different sites. So at first glance we can say, oh, this is shocking. Those sites coming from the fake contaminated river look like they have a higher mercury concentration than the other rivers. Who would have thought? Maybe we want to do an ANOVA and compare those different groups. So now we can do an ANOVA. One quick comment on analysis of variance with R. If you are just doing a one-factor model, this doesn't matter. But if you're doing something like a two-way ANOVA or anything with multiple factors, you need to realize that this analysis that I'm giving you now will just give you a type 1 sum of squares. If you have multiple factors, you need to look into some other functions that use type 3 sum of squares. It's not difficult, it's just a different function. And here are the functions that you would want to be using if you want to do use multiple factors in your ANOVA. Okay, so if we want to do an analysis of variance, we can use the function AOV, which is analysis of variance, and we need to give it information on our model and where the data is coming from. And we use that to create a model and assign it to a variable. So here I'm creating a model looking at the variability or how total mercury standardized is dependent on site in the data fish data. And then I'm assigning that to the variable mercury model. So I'll run that. And now I can look at a summary of the mercury model. And that will show me that the null hypothesis, which is that there is no variation in total mercury by site, is something that we can reject because our p-value is quite low. So here R will tell you, will indicate with the asterisks what the level of the p-value is, which is just a quick way of assessing whether or not it's significant visually. So if we want to reject the null hypothesis, the first thing that we need to do is make sure that our model is reasonable and that we're following the assumptions. So then we need to check the normality and homogeneity of the residuals. And then the next thing that we'd want to do is a post hoc test to see which sites vary from each other. So there are a bunch of different ways you can look at the normality and homogeneity of residuals, but I just wanted to show you that you could do it in just a couple of lines this way. You can look at the residuals versus the fitted values and see that they look all about like they're covering the same range, which is good. You can also assess normality with a QQ plot and add the line and say, okay, well, there's a little bit of variability. It might be because we're working with a relatively small data set, but generally it looks like it's probably normal. So that means that we can accept this model and move forward with the post hoc test. It's really easy to implement a two-key test. If you want to look at the help file for this function, two-key HSD, it will tell you about the two-key honest significant difference post hoc test. But basically, if you have a simple ANOVA, all you need to do is pass it that model to look at the post hoc test. So if we run that line, we see that the test is set up for 95% confidence interval. It will give you information on the model itself that you have passed it and what that model is. And then we can look at the pairwise comparisons and see, given the p-values of less than 0.05, the Cannificasis River is different from the contaminated river, St. John Harbor is different from the contaminated river, but the Cannificasis and St. John Harbor are not significantly different from each other. So I realize that all of that was a lot if you are an R beginner, but I'm hoping that that gave you a sense of how you might set up your analysis. So one thing to note here is that this whole file is 70 lines with all the comments. It's like maybe 15 lines of actual code. And that's all you need to be able to go through that sort of basic analysis of putting your files together, coming up with a standardized variable, and then running a statistical test. But by doing this in R rather than some other way, your data organization, your manipulation, data manipulation, statistics are all in the same place. Joining files is easy and automatic as opposed to full of potential pitfalls and mistakes. It's also easy to update your analysis with changes, and it's easy to update your analysis with more data. For example, if you have more sites or if you want to do another sampling year. So the next file, the next script that I'm going to show you will expand a little bit on that, how you can apply an analysis to another site or sampling year. So go back to your scripts folder, and you can open up the nitrification calibration R file, put that into RStudio. So this file was set up to work for another fake experiment, which I'll call the multiple ecosystems nitrogen cycling experiment, because I'm a biogeochemist and I think a lot about nitrogen. The idea is that this processes absorbance data for the nitrification assay and outputs nitrate concentrations. So basically what you need to know about a nitrification assay, or this particular one that I'm referencing, is that it's something where you have samples that change different colors depending on the concentration of nitrate, and then you can read the sample as the absorbance of wavelength of light at a certain wavelength. So you have different absorbances, and those absorbances correspond to different concentrations of nitrate. So it's just a standard linear calibration where you want to look at the relationship between absorbance and nitrate concentration in standards, and then use that to figure out the concentrations of your own samples downstream. So in this file, the script has the directions or the commands for reading the absorbance data, plotting a calibration curve to a PDF file, fitting a linear regression for the calibration curve, using that linear regression to calculate nitrate concentrations from absorbances, and then writing the sample concentrations to a CSV file. This one I'm going to be moving through fairly quickly, and there are a couple of concepts that I want you to pull out of it. One of them is that I want to demonstrate how using functions can help you create analyses that can be more easily applied to multiple data files. Another thing that I want to demonstrate is the use of code of this computational programming approach to be able to easily output both figures and data files, like CSV files, in ways that will also make your work more efficient. So again, as with the other script, we start by loading a package. We've already loaded this in this R session, so we don't need to run this, and the working directory is the same, so we don't need to run this line either. So the first thing that I'm going to do here that's different is I'm going to create a variable called nitrif data file name that has information on the file name that I'm going to read in first. And the reason I'm doing this is to more easily be able to change that file name downstream. So I'm going to start by walking through this code using the nitrification absorbances file. It's nitrificationabsorbances.csv. But then at the end, I'll show you how you can just comment out that line, comment in this other line, change the file name, and run the exact same thing on a similarly formatted file, and end up with the same kind of output for a different set of data. Right, so first we have to actually run that line. And now we have a variable called nitrif data file name. If I look at the value, it's nitrification absorbances as a character string. The next thing I'll do is read in my data. And here I'll use that same read.csv function. I'm telling it to find the file name by pasting together the data folder, this file name that I've created, and the .csv suffix. And then I'm assigning the value of the data to a variable called absorbance data, and then I'll be working with that variable downstream. So run that line, and now we have absorbance data, which has a couple of variables. One of them is type, which separates standards and samples. One of them is sample ID, which is only relevant for the samples and not the standard. We also have standard concentration, which is relevant for the standards but not the samples, and absorbance. So these top samples, the standards, are what we'll use to find the relationship between absorbance and standard concentration. So then we can apply that linear regression model downstream to the other absorbances for the samples. So the next 13 lines of code here are a function that I wrote. I'm not going to walk through all of it, but basically what it does is it plots the absorbance data for the standards. It fits a linear regression, and then it outputs an R² and plots it on the plot. So run all of those lines from 36 to 49. And then now we have a function called plot absorbance data that we can use to apply to further data. So I'm going to apply this function, plot absorbance data, to the data that I read in earlier, absorbance data. And then I have a plot that comes up in my plot window in the bottom right. So in these 13 lines of code of writing a function, I now can very easily plot data so that I've got absorbance on the x-axis, standard concentration on the y-axis. I've added this red linear fit and also an R² value to the top. So you can kind of see how having this function could be really handy if you're working with a lot of analyses. You can really easily go through them and just check all of the R² and also look at the figures immediately. So the next couple lines here are plotting that data, or plotting this plot to a PDF file. If you have a Mac, you'll be able to run these lines directly. If you have a PC, the quartz function here is one that talks directly to, or specifically to, a Mac plotting device. So if you should write a comment here, if you have a PC, look up the function PDF. So I'm sorry, I am including a non-cross-platform function here. But if you have a Mac, you can run those lines. And what it does is it creates a file in your figures that is a PDF that gives you this figure. So you can already, again, start to see how you could output many of these figures for different data sets and then just go through the figures and figure out which ones worked and which ones seem like they have a problem. So the next function that I wrote here uses a linear model to calculate, to fit the calibration linear equation, and then it creates a new data frame that has samples and concentrations. So I'll run the function, which just means defining it or creating it. And then I can call the function again on this absorbance data that I created earlier. And what it does is it gives me output of my sample IDs and also absorbances and concentrations. So it took that calibration that I created earlier and it applied it to all of the absorbances and then outputted the concentration. So here's my, my organized data. Here's the point where I could write this data to file and then read it back in to do analyses on the concentration, or maybe join this file to one that has information on the other samples so that I can start to set up groups and figure out whether there are differences among treatments. So once I've created this, I can then run a function to save the output to a CSV file, and again I'll use the write.csv function, run that, and it creates in the data output folder a file called sample concentrations nitrification absorbances. Okay, so one thing that I want to point out is that I created this nitrif data file name up at the top, and then I used that file name in a couple of different places. The first place I used it was when I read in my absorbance data here. So that file name determined what data I was working with throughout the whole file because it used it to read in the data. The next time I use that variable name is when I'm creating a plot. So I tell it what the file name of the plot should be based on that same file name variable. And then the last time I use it is when I'm saving the output to a CSV file. So I tell it, set the file to be something that's using that file name again. And there's a reason why I'm doing that. At this point, I could change this file name. So I'll comment out that first line, and I'll remove the hashtag from this second one, nitrif data file name. So now I'm setting up my file name to be nitrification absorbances year 2. And now if I run my whole file with the source button, I get a new figure here. And if I go back to my files, I now have two data files, CSV files, that have the absorbance data and the calculated concentrations. And I also have 2 different PDF files, one for each of those analyses. So that's how you can start to think about setting up your analysis in a way that you can apply it to multiple different data files, potentially from different years of data or different runs of data or different sampling sites or sampling dates or whatever makes sense in your own context. Okay, so I'm going to wrap up by explaining some best practices for when you're working with R, and then give you a couple of points on resources and places where you can get help as you're learning to use R. First of all, that self-contained project organization that I explained at the very beginning is a really good idea. It helps a lot when you're working with R. It makes it easy to find all your project files and to share your full project with other people. And then really importantly, it means that your relative file paths will work on any computer. So as long as you set your working directory properly, anything further downstream will be in reference to that working directory and will allow you to share those scripts more easily. Another key point that I cannot overemphasize is comment extensively. You will forget what you did later. It will not make sense. It's really helpful when you're using a function for the first time. You can add comments that are things like the digits argument does this, so that when you're looking at it later, you remember why you said it that way. You can also do things like, as you update analyses, at the top of your file, put in a comment with the date and say, on January 1st, 2016, I changed this analysis to have a logarithmic fit instead of a linear fit, and then you have a record of everything you've done. test things in the console but work in a script. So you can test out how functions work and which functions you want to use in the console. But when you're actually working with your own data, and it's an analysis that you'll need to repeat, or at least remember how to do, work in your script so you can save it. And then after you're done, clean up your code so that it all makes sense as a workflow. Be careful about your objects that are in your environment. It's not a good place for long-term storage. Make sure that anything you're working with is either something that can be recreated by a script or that's read in from an external file. This is really important because as you're working, you can start to create lots and lots of different objects just to test things out. But if you accidentally reference one of those objects in your R script and then it doesn't exist the next time you're running R, then you have totally lost that. So make sure that you can recreate all of your objects in your scripts and that you write your output to file, both in terms of data and figures. And of course, these suggested best practices have analogs for analysis outside of computational programming. So self-contained project file organization is a good idea no matter what you're doing. You may not be able to comment, if you're working in Excel, you won't have that really simple approach to commenting, like you don't have a place to put hashtags at the beginning of your file. But it's still a good idea to take notes on how you ran your analysis. It's just that it's sort of facilitated by the script structure of R. And last, here are some places where you can find help if you're working through R. As I showed you earlier, you can search help files for a function using a question mark. You can always Google your problem, be as specific as possible, include the term R. As you're Googling, if you see results that include Stack Overflow, that's a really helpful place to go. You can go to the R section of Stack Overflow directly, or you can look for it in Google search results. It's basically a website where people post questions and then other people respond with answers. And then the answers that are the best answers get upvoted, and so you end up seeing the most useful ones at the top. There's also an R help mailing list. If you're going to post that mailing list, look at the posting guide first. People respond very quickly. It can be rather dry. And if you accidentally use the wrong word for something, you may end up getting a response that has to do with your terminology rather than your actual problem. So just be careful if you use it so that you get what you want out of it. There are a lot of really great cheat sheets online. You can Google to find some. RStudio has a cheat sheet page that's super helpful. And last but not least, I'd like to plug my workshop that I'll be teaching over 2 days through the CRI, January 12th and 13th, where I'll go through a lot of these, I'll go through a lot of basic concepts in R and give you a lot of chances to practice different concepts so that you can really get up and running and be able to apply these concepts to your own data. And with that, I will take any questions.
Excellent, Lindsay. Thank you so much. That was a great presentation, so much good information, and a wonderful introduction to R. So as Lindsay said, we'll now open up the question and answer session. So you folks have 2 options for asking questions. You can either figuratively raise your hand and we'll unmute your microphone so that you can ask your question to Lindsay directly, or you can type in your question and we will read it aloud. So I should also mention that we are recording this presentation. I know there has been a lot of material presented, and it should be available on the Foundation's YouTube channel later on this week. So if you just Google YouTube ASCF for Atlantic Salmon Conservation Foundation and CRI, you'll be able to find our channel. So it looks like we've got some questions already.
Awesome. Hopefully I can answer.
Our first question is from Famida Hook, is her last name, or his last name. How can we participate in the CRI workshop, and is it free?
Those are both great questions. Registration is through Sarah Chuziak or through the CRI training. Darla, maybe we could email information on that out after the webinar for everybody who's participated. I'm not sure yet whether it'll take place in Fredericton or in St. John. I believe it's free, but don't quote me on it. It might not be. I don't actually know. That's information that I should know. So yeah, so we should email information on that afterwards so that people can access it.
Yep, we can definitely do that.
Great.
A comment from Melissa Palioras. She says, just a comment, great presentation, thank you.
Thank you. That's awesome.
Andrea Kirkwood adds, that was a terrific presentation. I would add that Twitter uses a hashtag R stats for crowdsourcing help from our users.
Oh, that's great. I'm going to add that to my list for presentation.
Okay, we have a comment from someone named Ol Olayemi saying it was a great presentation, but I think you move very fast, that we cannot move at the same pace because English is not everyone's second language. So I would reiterate that we are recording the presentation and it's free and it'll be available on the YouTube channel within a couple of days. So one of the great things about recorded presentations is you have the option to pause or go back and review anything that perhaps went too quickly the first time around.
That's absolutely fair. As I was developing it, I realized that that was definitely a trade-off. It in my workshop, I move much more slowly and give everyone a chance to have a lot of practice, and that's definitely a better structure, a better forum for being able to pick up these concepts for sure. So it's always kind of a balance to figure out how do you balance like the real learning, the real hands-on practice and learning with being able to give an overall picture of what those where you want to try to go, what that looks like. So yeah, trying to balance
Um,
between the two.
a comment from Richard Olwa saying, great presentation, and I'm looking forward to participating in the CRI workshop.
Oh, that's cool.
A question from, let's see here. Ooh, I just lost it. A question from Sheikh Zubar Hossein asking, I have graduated as an environmental engineer recently. I have not used R before. Would learning R help me in my future career?
Mhm. That's a really
As
good
I'm wondering
question.
about, mm,
Wondering, I would guess that, so I don't actually know enough about engineering to want to say that I have the final word on this for sure. But my guess is that if you're going to do anything with data analysis, that learning some sort of computational programming would be helpful. That's my assessment of related job searches with environmental science and also a little bit with data science. It could be R, it could be Python. Those are the two biggest ones that are used for science. Um, my guess would be yes, but you'd have to talk to somebody who does engineering specifically to confirm that. It certainly won't hurt. It might be so often I find that that approach is required for more technical positions, but it's really variable.
um, comments from a couple of different webinar presenters saying that they did find the pace fast, but they appreciated because it covered a lot of material and that they can always go recording to slow things down and watch it over again.
Sounds like there's a consensus.
Okay, and
That's okay. I'm used to
yes.
being told that I can't do that.
No, no, I think it just, a lot of material, especially for folks who are brand new to R.
Sure.
And I think that is one of the benefits of being able to record a presentation like this is that it then becomes a resource and a tool for people who want to continue to use R into the future.
Sure. I want to reiterate that what I really hope, so in that balance, I wanted to be able to show a lot of material, but I really hope that it doesn't end up being intimidating. So even though I was going through these concepts really quickly and they may have seemed sort of opaque in that context, when you take the time to slow down and practice with them, they're not, nothing is difficult when you can break it down into enough steps. So I know for someone who doesn't have a background in coding, it all feels like Greek, unless Greek's your first language, and then it feels like something else. Um, but it really, I just want to emphasize that it's really not that bad, and it actually starts to be kind of fun because even something as simple as like how do I set up the logic to be able to join these files is like a little challenge, and it's just a question of learning the vocabulary. And when you start speaking the language better, it starts to kind of start to flow and to feel a lot better and work a lot better.
I've got a question from Jun Yu Key, I think. Can I input raw data from sampling in R directly instead of through Excel and save it in CSV?
I don't think I know what you mean about raw data directly. R is able to read in a whole bunch of different kinds of files. Generally, things work best if they're in what's called a flat file, so something that's just two dimensions of column names, which are variable names, and then rows, which are your observations. So if that's a CSV or a tab separated um, file, anything that's like tab delimited or any other delimiter, I think is the word, you should be able to read it into R directly. So I just often find that it's easiest to open something in Excel as a CSV or like input into Excel as a CSV. But any output of anything that's a two-dimensional file with a consistent delimiter, you should be able to read into R directly. I think if you want to ask a follow-up question, I can maybe answer that better.
Thank you. Um, Andrea Kirkwood asks, a few academics have had to retract papers or make corrections to published papers due to errors when using R. Can you comment on how novice users can avoid the pitfalls of errors in code or use of the platform?
It's an interesting question. I'd be really curious to know whether those. Well, actually, as a general thing, I'm curious whether that's how that compares to people who have made errors not using R. I think it's like anything where you need to make sure you know what you're doing in every step and check your work for errors. I mean, I myself have done things where I was converting units and accidentally multiplied by 60 instead of divided by 60, something I had to do with minutes and hours. And you can't, no, it doesn't matter what platform you're using, you still need to be careful and check your work for errors like that. I guess if you're a novice user, based on my own pitfalls from my code when I first started, which was extremely inelegant, I would say that some of those best practices that I mentioned are really key. So make sure that you're not creating things in your environment that you can't recreate. So clear your environment before running a file to test it. Keep your code as neat as possible. It's okay if it gets messy when you're creating it, but take the time to go back and reorganize it, put in those comments, and make sure every line makes sense and is doing what you want it to do. And I mean, like any tool that you're working with, if you don't know exactly what it does, it's good to check with somebody. So especially if you're doing like I don't know, a statistical analysis or a multivariate analysis where there are a couple of arguments and you don't know exactly what they mean, it's good to check with somebody who does know what they mean and just make sure that it's doing what you think it's doing. Because I mean, ultimately, the computer will do exactly what you tell it to do, but it just may not be exactly what you want it to do. And so just making sure that what you're telling it is what you wanted is, I think, the key thing there. So yeah, so some combination of like checking your work, talking to other people. I mean, usually, I don't know, I find that our users tend to proselytize and really like helping other people with R. And just trying to keep your work as organized and sort of as clean as possible would be the main recommendations. But I mean, those really are things that apply to any approach, whether you're using R or Python or whether you're using like Excel and some other, I don't know, any other statistical program.
Thank you. We've got time for one last question. Melissa Paliuras asks, where can we get more information about Ladies Learning Code?
Oh, I'm so glad you asked. Okay, so Ladies Learning Code is a really cool nonprofit program. It started in organization. It started in Toronto and it's branched out. There, you can go to the Ladies Learning Code website. If you're in New Brunswick, we have a Ladies Learning Code NB Facebook page. I think there's a Ladies Learning Code regular Facebook page as well. I would start with their website and then move out from there. But there are a whole lot of really cool communities. They do a lot of workshops. It basically is a way of creating a comfortable space that encourages diversity in coding. Right? So like men are absolutely allowed to attend the workshops. They say we encourage you to bring a female learner as well, but it's not meant to cut out different groups. It's meant to sort of make everyone feel more comfortable where they can learn in a comfortable environment. So yeah, check out the website. They're great.
Excellent.
I'm happy to plug them.
Thank you so much, and thank you for an excellent presentation. We had a lot of folks sign up, and I think a lot of folks will be watching the YouTube channel afterwards.
That's great.